Welcome everyone to join the online seminar of the Science of Childhood Cancer. Today is our great pleasure to be able to have Dr. Aviv Grigrev, who is the Executive Vice President of Genentech, to give us a wonderful seminar at the Science of Childhood Cancer. So many of us who know Dr. Grigrev is based on her outstanding work while he was at the Broad Institute, where she served as the chair of the faculty and core institute member, as well as the founding director of the Common Cell Observatory at Broad and MIT. She has really pioneered a lot of the outstanding competition tools. In particular, her lab has led the development of experimental approaches as well as analytical approaches for analyzing single cell data. And she is a professor of biology at MIT and investigator of Howard Hughes, and also a founding co-chair of the Human Cell Atlas project. Dr. Grigrev served on multiple corporate advisory and also a scientific advisory board for St. Jude Children's Research Hospital. And she has been a leader in deciphering molecular circuits that govern the cells, tissues, and organs in health and their malfunctions in disease. And she is a member of the National Academy of Science as well as National Academy of Medicine. And she's also a fellow of the International Society of Computational Biology. So we really look forward to her lecture today that will tell us more about the single cell atlas.
Thank you so much. Let me just share my screen, which I can do now. It's a real pleasure to be here. It would have been so much fun to actually be in St. Jude's in person. It's one of my favorite places. And as Jingui said, I'm actually a member of the SAB, and it's one of the things that I do with particular joy. Maybe by this fall, it will be possible again, but it's so nice to see everyone virtually as well. And so I'm going to talk today about cell atlases as roadmaps in cancer, and I'm really grateful for this opportunity. I'll start with my disclosure. I'm a co-founder and equity holder in Celsius Therapeutics and in Immunitas, and until July 31st, I was an SAB member with cash or equity compensation in Thermo Fisher, Neogene, and Syros. From August 1st, as Jingui told you, and I'm an employee of Genentech, which is a member of the Roche Group. All the work that I'm showing today was still done in my lab at MIT Broad and HHMI, even though the lab is gradually moving to Genentech. And so I'm going to talk about single-cell genomics today, which has been instrumental in building tumor atlases. And the hope of those atlases was that it would help us understand tumorigenesis, drug resistance, and metastases. And today, I'm going to present several vignettes on how the high resolution of single cells in tissues and beyond helps us understand the human biology of tumors. And I'll first share two related stories on understanding immune exclusion in tumors, starting with melanoma and then moving to synovial sarcoma, which is a childhood cancer. And then I'm going to turn to two other vignettes that focus on new methods that we hope to use to dissect the control and the functions of different kinds of programs in tumor cells and their impact on the microenvironment. For that, I will turn back to melanoma, that has proven to us a really great starting point for many studies in other cancers. And so I'm going to start, as always, with a patient tumor, and in this case, with melanoma. Now, as you all know, many melanoma patients have benefited tremendously from checkpoint inhibitor therapy, and other patients, unfortunately, have not. And that could be due either to intrinsic resistance that existed prior to therapy or due to acquired resistance. One of the major hypotheses in the field is that resistance, at least in some patients, is associated with what we call cold tumors that don't really have infiltrating T cells, whereas the tumors that are more responsive are hot and they have tumor infiltrating lymphocytes, especially T cells. And so we wanted to begin and gain some insight into this. And for this, we looked at single-cell RNA-seq data that we acquired from 31 patients. Half of them were treatment naive, and the other half were after treatment, and all of those after treatment were non-responders. This was not, the nicest, most matched cohort that one could put their hands on today, but at the time that we started this study, which actually traces back to 2012, 2013, these were very early days for single-cell RNA-seq, and we really had to make do with what we had. And so focusing on malignant cells, you can see them in the top plot. The cells are colored by the patient, and you can see that the tumors are very different. So each patient's cells separate from each other. And that's a common pattern that we observed across many cancers by now. But we still want to see whether we can find some common features. So for this, we devised a new computational method that combines single-cell RNA-seq from a few patients, like we had, with bulk RNA-seq from many patients in TCGA. And of course, in TCGA, all the patients were treatment naive. There were no treatments, in particular, there were no immunotherapies at the time of TCGA. So from the single-cell RNA-seq data, we start by identifying genes that are specific to T cells in tumors. And based on those genes, we use this as a T cell specific signature to infer the level of T cells in TCGA profiles. Then, using these levels, we search for genes that we know are specific to malignant cells from the single-cell RNA-seq, but whose expression in the TCGA tumors correlates positively or negatively with either a high or a low proportion of inferred T cells in the bulk tumors. Then finally, this forms a seed for us. We know these genes are specific to malignant cells because of the single-cell RNA-seq. We know they track with T cell levels in the bulk tumors because the signatures for the T cells were based on single-cell RNA-seq. But we want to see if there's additional genes in the malignant cells that go together with this seed, regardless of whether they are unique to malignant cells. And for this, we go back to the single-cell RNA-seq data and use the signature to extend and find genes that co-vary with the seed across the single malignant cells. And this gives us finally what we call a program. We call it the T-cell exclusion program because it's associated with T-cell exclusion. The repressed part of the program, which is lower in cold tumors and higher in hot tumors, has genes like MHC class 1 and interferon gamma and the senescence-associated secretory phenotype. Whereas the induced part of the program, which is higher in cold tumors, it has genes like MYC and CDK4 and CDK targets. And all I ask you to remember from this slide for right now, for later on, is the CDK4 and the CDK targets. I'll come back to that. But first, we wanted to confirm that this was a real thing. This was all statistical inference. And in order to confirm that this program is really associated with exclusion, we went in situ. And so we used a new method that was developed at that time by Peter Sorger's lab, T-Cisive. And in collaboration with Peter's lab, we measured 13 proteins in FFPE sections from 19 of our patients. And those are the genes that mark the key cell types and the resistance program. And so to orient you, here's just one section. In blue are the T cells. The CD8 plus T cells are in light blue. Red are malignant cells that repress the exclusion program. So we expect them to promote a hot environment. And green are malignant cells that induce the exclusion program. So we expect them to promote a cold environment. And this is exactly what we see. You can see that the red malignant cells are really interlaced with the blue TILs, but the green cells make these big cold patches. And this is actually true across the patient samples. And we can quantify it statistically to show that the exclusion score is significantly higher in malignant cells that are in cold T-cell depleted niches. Okay, so now we believe that this program is a real statistical signal of association, but we want to understand it more. So we look at how this program behaves in the cells in each of our patients' tumors where we had single-cell RNA-seq, looking both for variation within a tumor and variation between tumors. And when we look at the cells from untreated and resistant patients, we see that some of the cells from the treatment-naive patients already express the programs. So this might reflect an intrinsic resistance mechanism. Now, we couldn't test it directly on the original cohort. It wasn't ever designed to allow us to ask questions like these, but it was good at raising hypotheses. So instead, we tested this in a bigger cohort of 112 patients in collaboration with Dirk Schandendorf in the University of Essen in Germany and Ellie Van Ellen at the Dana-Farber, where the patients were biopsied prior to checkpoint inhibitor treatment, and we had bulk RNA-seq from these pre-treatment biopsies, and then the patients were monitored for outcome. And what we found was that the signature expression in bulk tumors before treatment was predictive of both progression-free survival and of clinical benefit and the duration of the benefit. Okay, so next we wanted to see if there's drugs that may revert this program, acting on malignant cells, because this program is expressed in malignant cells. So for this, we queried for drugs that are more toxic to cell lines that overexpress the program than for cell lines from the same tumor type that do not express it. And the third top hit was a CDK4 and 6 inhibitor. And you might remember that I told you to remember that CDK4 and CDK4 targets were members of the induced side of the program. So that makes very nice sense for us. and we decided to test this prediction. We treated melanoma cell lines first with the CDK4 and 6 inhibitor abemaciclib, and then we profiled them with single-cell RNA-seq before and after treatment. Before treatment, about 10% of the cells express the programs, which we see in purple. And after treatment, this proportion is radically reduced. And in addition, the remaining cells induce the downside of the program, especially the SASP. And I want to point out that we don't think that this is about cell killing of the cells. This is actually about shifting their cellular states. And we further tested it in patient co-culture with autologous TILs. I'll come back to this system far later in the talk. And so then we turn to a B16 melanoma animal model, which you might know is typically resistant to checkpoint inhibitors. And indeed, if you treat it alone with an anti-PD-1 or in combination anti-PD-1 and anti-CTLA-4, you don't really get much of a response. But we found that combination therapy where we add abemaciclib after the initial checkpoint inhibition led to substantial benefit. And we showed, even though I'm not showing you here, that this requires having RB-competent malignant cells, which means it is on target, and on the presence of CD8 T cells. So let me summarize this first part. I showed you how we use single-cell genomics to make a statistical prediction about the program in malignant cells that leads to T-cell exclusion, that is associated with, we didn't have causality, associated with T-cell exclusion in the tumors, statistically. Then we validated it, that this association also holds in situ. After that, we validated that it predicts the response to immunotherapy in 2 independent cohorts. I showed you one of them. There was a second cohort with the same result coming from MGH. And then we used additional computational approaches to predict that it could be reverted by CDK4 and 6 inhibitors. And we validated this in cell lines, in co-cultures of patient cells with autologous TILs, and in animal models. Great. So next we asked, what might happen with other tumor types. So we decided to take our program and score it across multiple tumors in TCGA. We didn't have single cell data for each of these tumors, so this is not as clean of an analysis as we can do in other settings, but the results were quite suggestive. Cutaneous melanoma, lung, adeno, and kidney all have relatively lower scores, and this is consistent with the benefit of immunotherapy in melanoma. On the other hand, if you look at things like uveal melanoma or colon cancer, they have relatively high scores. And the highest score was in synovial sarcoma, which is a childhood cancer that I'm going to turn to next. And so a little bit about synovial sarcoma. It's an aggressive mesenchymal cancer. It accounts to about 10 to 20% of all soft tissue sarcomas in young adults. Synovial sarcoma tumors are driven by an SSAT-SSX fusion protein, where the BAF subunit SS18 is fused to one of 3 SSXs, either SSX1 or SSX2, which are the 2 common fusion, and rarely SSX4. This results in reprogramming of the chromatin landscape, of how the BAF complex basically regulates chromatin. And it leads to a range of malignant cell phenotypes. And one of the interesting things about synovial sarcoma is that these are tumors that homogeneously express very immunogenic antigens, known as cancer testis antigens or CTAs. And they are recognized, these antigens, by circulating T cells in the peripheral blood of patients. But at the same time, T cell infiltrations into the tumors is exceptionally low, and they don't benefit from immunotherapy. And so this was consistent with our signal of our melanoma program being highly scoring, our exclusion program in synovial sarcoma. So it gave us the appetite to dig in more. And for this, we assembled a single cell atlas of 16,000 cells from 12 resections in 10 patients. This is a large number because the tumor is rare, and even bulk genomic studies in this domain are pretty small. And in the single cells, as I showed you before, we can readily distinguish between malignant and non-malignant cells. We do this actually by inferring copy number variants, as well as by detecting, in this case, the fusion transcript that we can see directly in the cells. And then the malignant cells, which you see on the left, are separated by the patients, as we typically see in all tumors. I showed you that in melanoma too. But we can also distinguish within a patient between epithelial-like and mesenchymal-like cells in the same tumors for tumors that have had biphasic pathology, which are expected to have 2 different kinds of cells. Now, if I look, on the other hand, on the non-malignant cells, which are plot 2 in this slide, and now we're focusing on them, we can see that they mostly separate by cell type. And so cells of the same type are grouped together across the patients. And that's, again, a phenomenon that we see repeatedly in single-cell RNA-seq of tumors. And so now we turn to ask what might be the properties of these cells and how could they maybe explain the coldness of the tumor. For example, maybe there's a cell there, but they're not all that as far as immune cells go. And here we had somewhat of a surprise compared to our expectations, because the immune cells in the synovial sarcoma microenvironment in these tumors that were profiled appeared to be quite active. So for example, these are macrophage states, and they were very comparable to the macrophage states that we see in melanoma patients. We see both M1 and M2 states, so pro and anti-inflammatory. And if anything, we see higher levels of the inflammatory gene program. Then, my slide stopped advancing. I hope they're still advancing. Then when we turn to CD8 T cells, we found both singletons and expanded clones. So we found 57 clones, they were all patient specific. When we had two paired samples for the same patient, those had shared clones. And then we could take the expanded and the non-expanded, so T cells from expanded and non-expanded clones, we compared them one to the other, and from this, we derived a T cell expansion program. And so now we looked at the behavior of cells that highly express this program in terms of the level of cytotoxicity gene expression and exhaustion gene expression. And we found that cells that express the T cell expansion program show higher cytotoxicity compared to exhaustion score. Usually these two scores are very highly correlated in cells. And so it seems that we have nice expanded T cells, they're more cytotoxic, and they're less exhausted. And still, the immunotherapy is not beneficial in these patients. And so then finally, for programs like effector versus exhaustion scores or responsiveness to checkpoint inhibitors, the CD8 T cells from synovial sarcoma actually scored higher than the CD8 T cells for melanoma. And so if anything, CD8 T cells in synovial sarcoma are more active, more expanded, and seem to be more responsive than those in melanoma. And yet, immune cell infiltration is especially low. And so if you use our single-cell RNA-seq profile of synovial sarcoma now to infer immune cell infiltration levels in each of the tumor classes in TCGA, similar to the analysis I showed you in melanoma earlier, synovial sarcoma is the lowest. And it's also significantly lower than other sarcomas. So it's not the intrinsic states of the immune cells. Now, of course, that makes us think, well, what are the malignant cell programs doing instead? And so for this, we turn to look at the malignant cells to try and find shared features. As I told you, the cells primarily separate by patient. And so if I did a simple analysis, like clustering and differential expression, it's not going to work. It's going to emphasize differences between patients rather than differences within patients or commonalities across patients. So instead, we used non-negative matrix factorization, NMF, a very robust decomposition method, which lets us find programs that may vary both within and across different patient cells. And we recovered 3 programs. One is a program of epithelial versus mesenchymal differentiation. And so it distinguishes epithelial and mesenchymal-like tumors, but also epi-like and mes-like cells in the same biphasic tumor. The second is the cell cycle. It scores some cells within each tumor. You can see them in these little red dots. But, okay, so it scores some cells in each of the tumors. And then, though, we had a third program, and that was the most surprising one. This is a program that varied within the cell of each tumors. Okay, so there were some cells in each tumor that highly expressed this program and not others, and this pattern repeated itself in every tumor. And this was done in a way that was separate from the differentiation or the cell cycle patterns that we found with the other programs. Now, because each tumor had some cells with this program, we decided to call it the core oncogenic program. It induced genes that, the induced genes in this core program were enriched for oxidative phosphorylation and for carbon metabolism functions, and the repressed genes were enriched for TNF signaling, apoptosis, p53 signaling, and multiple tumor suppressors. And we validated its expression in a subset of tumor cells in each of multiple tumors in situ, and also we found that it overlapped significantly with the genes of that exclusion program that we had in melanoma. Now, each of the programs had prognostic signal, the low expression of the differentiation program or the high expression of the core oncogenic program in bulk RNA-seq were each independently of each other associated with poor survival. So now we asked, could there be a relation between the core oncogenic program and the immune microenvironment? So the first clue that there might be came from a partial correlation analysis we did to relate the inferred proportions of different microenvironment cells with the level of the program after conditioning for tumor purity, which otherwise would be a massive confounder. So when we do this, we find that the core oncogenic program is negatively correlated with the immune content of the tumor. And this is in 2 independent studies. Now, we wanted to establish this correlation further. And again, the ideal approach to this, of course, would be to look directly in space. So we turned to a spatial transcriptomics analysis. And here again, we relied on a relatively new method from NanoString called the GeoMX DSP. In this method, we can use FFPE tissue again. We stain with proteins of interest, and we first choose what we call a region of interest, or an ROI. And then within each region of interest, we can use a mask in order to capture only the RNA from a particular area of illumination, or AOI, within the ROI. And so in our case, we stained tumor sections with markers for immune cells, CD45, and for epithelial compartments using the pan-CK set of markers. From these, we define immune, which is CD45 plus, and non-immune, CD45 minus cells. And then in the CD45 minus cells, we can distinguish between malignant cells by cytonuclear atypia, and then further as epithelial-like, which would be pan-CK plus, or non-epithelial-like, which would be pan-CK minus. So from this rich information, we can now distinguish multiple ROIs in each sample, and within each ROI, we can distinguish multiple AOIs in each of the ROIs, so that we would separately profile in each ROI the RNA from the CD45 plus areas, from the malignant CD45 minus pan-CK minus, and from the malignant CD45 minus pan-CK plus ROI. So they're all in one niche, but 3 different sources of RNA. And overall, we had 306 spatially distinct areas that we profiled in this way. Now, what did we learn? We related our core oncogenic program score in the malignant areas, where we know we're literally capturing RNA from malignant cells, to the immune abundance based on CD45 plus in the same region of interest. And we found that there's an inverse spatial correlation. The higher the core oncogenic program is in the malignant cells, the lower the CD45 abundance. And the core program genes are prominent amongst the genes that are enriched in malignant cell AOIs from region of interest that have low CD45 plus abundance. And we tested, you should know, more than 9,000 other gene signatures, both general ones and ones that were specific to cell types and to tumors, and none of them showed this association except for the core oncogenic program. Now, correlation, though, is not causation. And so what we want to ask now, is it that the immune cells suppress the program, or is it that the program excludes the immune cells, or maybe both of these things are true, and there is some feedback loop between them. And so for this, we turned back to experiments. And we went to the malignant synovial sarcoma cells and wanted to see how this program, first of all, is regulated. And obviously, since this is synovial sarcoma, which is driven by a chromatin regulator, we started with a fusion protein. We knocked it down in each of 2 synovial sarcoma cell lines and followed it by single-cell RNA-seq. And we found that the fusion protein, oops, I have an issue with my animation again. We found that the fusion protein controls the program so that it is repressed in the knockdown. But at the same time, the fusion also represses the differentiation program, and both of these effects are independent of the cell cycle. And so now, by adding ChIP-seq data from our collaborator Sigal Kadosh's lab, who studies synovial sarcoma and the BAF complex, we could also show that the fusion protein directly binds and regulates the program genes. And that includes 4 prominent transcription factors, as well as CDK6 and CCND2. So there is a direct genetic connection. So now we're down to causality of what regulates the program, but we don't yet understand the niche. Now we ask, what about the immune cells? Could they also impact the program in malignant cells? For this, we first used a mixed effect model in order to associate the expression of ligands in immune cell types to the state of malignant cells, the expression state. And the strongest signal that we found was of interferon gamma expressed by CD8 cells and TNF expressed by macrophages. And in both of these cases, these expression patterns were associated with repression of the program in malignant cells across our tumors. We also found this association in the nanostring spatial data. The higher interferon gamma and TNF levels we saw in CD45 plus areas of interest, the lower the level of the oncogenic program was in the malignant AOIs in the same spatial niches. So now back to experiments. We turn to test this experimentally by treating the primary synovial sarcoma cell cultures with TNF and interferon separately and in combinations, and again followed by single-cell RNA-seq. And we found that the joint treatment with interferon and with TNF represses the program, although TNF alone only represses it temporarily. So now we want to put these pieces together, just like in the melanoma case, and suggest a therapeutic hypothesis and see if we can identify compounds that would repress the core oncogenic program in synovial sarcoma cells and induce more immunogenic cell states. So we first modeled a core oncogenic regulatory network where the SSX and SS18 HDAC1 complex came as the program's master regulator, and the tumor suppressor CDKN1A, also known as P21, was the most repressed target. And according to this model, the core oncogenic program regulates, but is not regulated by cell cycle genes through the P21 CDK246 axis. And this is on top of the direct induction of cyclin D and CDK6 by SS18 and SSX, which I already mentioned to you. Now, based on this model, we postulated that cell cycle modulators like CDK4 and 6 inhibitors and inhibitors of SS18, SSX like HDAC inhibitors should jointly target the immune resistance features of synovial sarcoma cells, especially in the presence of cytokines like TNF. Okay, so we wanted to test this. We treated synovial sarcoma cells as well as mesenchymal stem cells with abemaciclib, the CDK4/6 inhibitor, TNF, based on the signal from the immune cell, and binostat, which is an HDAC inhibitor. And we did this alone or in combinations, and then we performed single-cell RNA-seq. And we found that HDAC and CDK4 and 6 inhibitors synergistically repressed the core oncogenic program, the melanoma exclusion program, and the SS18 SSX program, and the induced MHC class 1 expression. So we're seeing joint effects, and not only that, they exceed the expected additive effect, suggesting that we have a real synergistic interactions. So now, of course, we wanted to ask if we modulate the synovial sarcoma cell states with HDAC and CDK4 and 6 inhibitors, would we be actually impacting T-cell mediated killing? And so for this, we took the synovial sarcoma cell line CME1, and we engineered it to express cancer testis antigen, a CTA, called NY-ESO1. And then we pre-treated the cells with HDAC inhibitors and CDK4 and 6 inhibitors, took away the treatment, co-cultured the cells with NY-ESO1 reactive human T lymphocyte cells, and asked what would happen. And what we found was that the combined treatment of the synovial sarcoma cells with HDAC and CDK4 and 6 inhibitors before co-culture induced MHC class 1 cell surface protein expression. That's consistent with our previous study. And then we saw increased T-cell activation in the subsequent co-culture, and that's reflected by higher CD25 expression on the T-cell surface, and increased secretion of interferon gamma and IL-2. And ultimately, we finally saw increased T-cell mediated killing, even though only the malignant cells and not the T cells were ever exposed to the drug combination, which is especially important with abemaciclib that can act on T cells directly. And so I showed you that even though synovial sarcoma is a cold tumor type, the limited immune cells that are present are active, and they have similar features to those in melanoma. And then in order to understand why the T cells are not effective, we looked at the malignant cells. We found that the malignant cells vary by their cell cycle, epimass differentiation, and a core oncogenic program. This core oncogenic program has shared features with the melanoma exclusion program, and it's associated with poor prognosis and with cold tumors and with cold tumor niches. And then we found that interferon from CD8 T cells and TNF from macrophages can synergize to repress the program. And on the other hand, the program is directly activated by the SS18-SSX fusion. And finally, we found that we can directly and synergistically repress this program by combining an HDAC inhibitor and a CDK4 and 6 inhibitor. And that leads also to enhanced T-cell mediated killing in vitro. Okay. So now, how can we dig deeper into programs like these? You know, the CDK4/6 thing was kind of a lucky strike that there was a gene and there is a reagent and there is a small molecule and it's even an approved drug. What we need, though, in a program, there's dozens and can be hundreds of genes, and we need model systems that we can actually manipulate in the lab. Just like the co-culture systems that I showed you that we use to assess the impact of CDK inhibition in melanoma or of CDK and HDAC inhibition in synovial sarcoma. But these kinds of programs, again, dozens of genes and no targeted inhibitor, and that actually means that we would need a genetic screening approach. And that introduces a whole set of challenges. First, we need to be able to assess the impact on a gene program, which, as you saw, is heterogeneous within the malignant cells. And second, the cell population that we have is going to be mixed because we have to run a co-culture experiment, for example, with T cells. So to address some of these challenges, we decided to use a method we developed several years ago called Perturb-seq, which combines a pooled CRISPR screen design with single-cell RNA-seq as the readout. And in Perturb-seq, both the perturbation guide or barcode and the cell's profile are recovered by single-cell RNA-seq. So we can do the entire experiment in a pool, and after sequencing, we can match each perturbation to the transcriptional profile of the cell from which it was expressed. But we had an additional challenge because many of the genes we're interested in, this is, for example, the melanoma case, especially those that are down-regulated in something like the exclusion program, they can be robustly detected at the protein level on the cell surface, but they're not well detected by single-cell RNA-seq. And you know, many also of the best established phenotypes are for cell surface proteins, not for RNA. So to look at both the RNA and the protein levels, we modified Perturb-seq to rely also on Site-seq, a method that allows us to measure proteins on the cell surface together with a single-cell RNA profile. And so we stain the perturbed cells by DNA barcoded antibodies, and then we perform Site-seq to have both RNA and proteins associated with our guides. The reference is actually outdated. The paper came out this week. Now we set to perturb 248 immune checkpoint resistance and exclusion program genes in patient-derived melanoma cells and assess the impact in two ways. One on cell viability in TIL co-cultures. This is a classical CRISPR viability screen. And the second is on cell profiles in co-culture. And we did the experiments with and without interferon gamma. So first, in the viability screen, the strongest hits were genes like beta-2 microglobulin, HLA, and the JAK-STAT pathway genes, which, when we perturb them, give resistance to T-cell killing, and that's a nice positive control. But there were also additional genes that confirmed resistance when they were perturbed, including CD47, CD58, and CDH19. So now we wondered, are these all working through the same or through different pathways? And for this, we turned to the Perturb-seq screens. And here we compared the profiles of 200,000 patient-derived melanoma cells from either controls or treated with interferon gamma or in co-culture under the different, first without the different genetic perturbation. And the cells partitioned by their treatment based on either their RNA profiles or their protein profiles. So first we wanted to know if the RNA and the protein phenotypes were at all relevant to our question. So we looked at the impact of each condition, irrespective of the perturbation. The data is from the Perturb-seq experiment, but we're not treating the guides yet here at all. They're coming in a second. And we're looking at the effect on the level of RNA and proteins. And we saw that many of the genes whose perturbation impacted susceptibility to killing in the CRISPR viability screen were also regulated at the RNA and the protein level, and often in both levels. So this kind of concordance shows us that expression levels are a relevant phenotype. Next, we wanted to determine the actual impact of each genetic perturbation. So we built on a computational model that we developed before called MIMOSA that uses the expression of genes across the cells and the identity of the perturbations per cell in order to fit a regulation matrix of perturbations or guides per cell. And we addressed several of the technical challenges that remain through the model, including estimating whether a cell had an effective perturbation by a guide, and testing for the significance of the associations. And the most prominent portion of the regulation matrix aligns with the viability screen. JAK, STAT, and the interferon gamma receptor inhibited a program of MHC and antigen presentation genes, chemokines, inflammatory cytokines, interferon response genes, and so on. Kind of all the usual suspects regulated by all the usual suspects. But on the other hand, their perturbation also induced genes like CD58, which I'm going to return to later. So again, this was a great positive control. The screen definitely worked as a biological experiment, but what else can we learn from the screen? So for this, we looked at the rest of the regulation matrix of the impact of the perturbations on RNA and proteins. On the one hand, we clustered the perturbations. And in this way, we identified functional modules, or call them co-functional modules, of perturbations that had a similar impact across the RNA and proteins. On the other hand, we clustered the RNA and protein profiles. And in this way, we find programs of genes that are co-regulated across the perturbations. And overall, when we put this together, this gives us 6 co-functional modules that impact 4 major programs. So for example, in module 1, all of SOX4, RB1, SP100, and IRF3 activate a program of cell cycle, metabolism, inflammation, and checkpoint genes. These kinds of regulators are themselves repressed when we perturb the JAK-STAT pathway. And also, SOX4 and RB1 inactivation was shown to impact response to immunotherapy in melanoma or breast cancer models. On the other hand, this same program is also repressed by module 2, which has genes like CD58 and CXCR4, which are themselves genetic activators of SOX9 and SP100. So now we have a full circuit of what is happening in the cells under these perturbations in co-culture, some of which might be directly related to our clinical phenotypes, and some of it is, the business of the cell being perturbed. And so one of the hints that we have a real clinically meaningful result is that the perturbations also change the expression of those exclusion program genes from which we started. So when we perturb JAK-STAT pathway genes, the program is increased, and perturbation of other genes like CDK6, MYC, ILF2, DNMT1, and ASCL3 repressed this program, consistent with what we learned about CDK4 and 6 inhibitors. So now that we have this large set of genes parsed out, can we use it as a map for distinct mechanisms that might be related to exclusion or resistance? And I'm going to illustrate this with one example that we focused on, which is CD58 that I already mentioned to you twice, but let me recap all the evidence we collected. CD58 was part of the repressed side of the exclusion program in patient tumors, meaning CD58 is down when in malignant cells, when tumors are T-cell excluded. CD58 knockout cells are more resistant to T-cell mediated killing in the viability screen. CD58 is not just activated by the JAK-STAT pathway. In fact, it's actually induced by JAK-STAT knockout. So it behaves opposite to other things that are typically regulated by JAK and STAT, not just the same genetic mechanism. The CD58 knockout has a different phenotype than JAK-STAT knockout in the perturbed Site6 screen, and it did not affect the expression of antigen presentation genes. So this together suggested to us that CD58, that the loss of CD58 is a distinct or a possible distinct resistance mechanism. So just as a quick reminder, CD58 is an adhesion protein. It's typically expressed on the surface of antigen-presenting cells, or APCs. It binds to CD2 on CD8 plus T cells, and not on on T cells and on NK cells. And it has come before in viability screens in the context of cancer immunotherapy, but there is very much less known about it, and that's partly because there's no known mouse ortholog for it. So you can really study it only with human cells and human data. And so we validated that CD58 knockout melanoma cells are resistant to both T and NK mediated killing. And we also validated the perturbed Site6 result that the CD58 knockout does not impact MHC class 1 or 2 expression, but it does induce, PD-L1, which may be one of the mechanisms by which it impacts resistance. And finally, And finally, we went back to the patient data and we saw that it's downregulated in patient cells after immunotherapy. And so to summarize this third part, I showed you how we can use perturbed site-seq to dissect a gene program of exclusion in melanoma. We parse out a well-established JAK-STAT module, but also 4 other modules that govern 8 programs, including the exclusion program. And one highlight is the distinct role of CD58, but there's many more other regulators and targets that we hope to further pursue. In the last 5 minutes or so, I want to show you one layer that we're digging deeper into now. This last part of the work is not at all published yet, not even posted as a preprint. And so this is when we wanted to start looking in the other side of what happens in tumors, which is not how their cell states are changing, but how their genetics is changing, as we've learned so much from SS18SSX, to think about the genetic perturbations as well. And so, what about this general backdrop of driver mutations that are associated in tumors? There's a very large number of mutations for tumors like melanoma, and I'm not talking about the background mutations. There's a large number of driver mutations too. And so understanding retrospectively from tumor which combinations of mutations matter and in what way can actually be quite difficult. And we want to really understand this. And so our question was whether single-cell genomics can help with that. And so several years ago, Eran Hodis, when he was actually a joint grad student in Levi Garraway's lab and my lab, and that was a while back, he's a resident now, he decided to engineer a system to allow us to assess this. And he started with healthy primary human melanocytes and sequentially built 9 genetically distinct isogenic models, hitting 6 common melanoma pathways: RB, the MAP kinase pathway, and telomerase, which together we call the core mutations, and then adding on top of them either P53, P10, or APC loss of function, and then finally adding either the P53 or the APC loss of function on top of P10. Now, I'm going to label the genotypes with these acronyms, CBT3, P, and A, and that would allow me to denote all of the genotypes. And we have cells with anything from 0 to 5 different precisely genome-edited mutations per cell. The 3 core, and by the way, that was a huge experimental effort on Eran and John Kwon, who was a research associate with him and now is a medical student. I'm just not describing to you the entire experimental saga that led to all of those cells. Now, the 3 core mutations together lead to immortal melanocytes. This only happens after we activate telomerase. But now we ask, what other characteristics do these cells have? And we start with characterizing the cells molecularly in vitro. And when we profile the cells in vitro by single-cell RNA-seq, each of the cells here is color-coded by the genotype. And you can see that the consecutive mutations actually produce an ordered transcriptional progression with a couple of major transition points, like the one that happens when you hit the 3 core mutations moving from CB to CBT when you activate telomerase. So now we want to dissect the underlying expression changes. And for this, we learned gene programs, similar to, with the same method, NMF, that we learned programs in synovial sarcoma. And we can see that one program can be related to more than one genotype. For example, we have an interferon program that's induced by the first 2 mutations and is shut off thereafter. We also have an EMT program that really marks the main transition through the 3 core mutations, and so on. We have a WNT program, a melanosome program, a MYC program, a TORC program, and the different phases of the cell cycle. Now we can take those cells that we know are phenotypically and genotypically different from each other, and we can assess the phenotypes that are associated with them in a mouse model by using NSG mice. So cells with the 3 core mutations together give these very small nodules in about 2 to 4 months, and then they make these very small melanomas 5 months or more in. And now we can look across all of the genotypes, and we can see how combinations of mutations are associated with distinct phenotypes, with primary tumor growth and at least some metastasis observed with any 4th mutations, and then pigmentation really only happening when we have the A or the P3, so APC or P53 combinations, and only CBTPA having really very aggressive disease. Next, we isolated and we profiled both the tumor and the mouse stroma cells from each of these tumors. And we're starting here with the cancer cells from the NSG mice in vivo, again colored on the left by genotype. And we see these substantial differences of the programs across the genotypes. You remember having seen them, the same types of separations in patient tumors. And we see a very nice agreement between the in vivo and the in vitro program. But what is even cooler is that the in vivo programs are actually consistent with programs that we derived from patient tumor cells, even if they were originally called by different names. And even more than that, the programs are actually associated with the same genotypes. And so, for example, both the patient-derived oxidative phosphorylation and our in vivo OXPHOS melanocyte programs, they're overlapping in their genes, but they are also both enriched in tumors with mutation in Wind pathway genes and a high level of pigmentations in our mouse model, in our NSG model with our human engineered melanocytes, and in actual patients with those mutations. And then finally, we turn to the stroma side, profiling tumor-associated mouse cells. Now, these are NSG mice, so there's no BT or NK cells, but we did see clear distinctions in the tumor microenvironments of tumors with different genotypes and ages. The most dramatic example is with neutrophils that are nearly 40% of the stroma cells in CBTA or CBTP3 tumors, but they're a very small minority in any other genotype, including CBTPA. And that really highlights how important it is to look at different combinations. And so some changes really happen with age, like the shift to an almost only contractile fibroblast in older CBTP tumors. The melanocyte genotype also impacted the state, not just the proportions of tumor infiltrating cells, for example, here for stromal cells. So M1 macrophages from, here it is for tumor infiltrating macrophages. So M1 macrophages from 2 months old CBTP tumors, they differ from CBTA and CBTPA tumors, and which have Wind pathway mutation, which might actually explain the source of the difference. And also in neutrophils, even though both the CBTA and the CBTP3 have high infiltrating neutrophils, they're not the same neutrophils. They do all span the full spectrum, but the CBTA neutrophils have a stronger program, which is typical of tumor infiltration, and the CBTA melanocytes actually express CCL2, which is a known neutrophil attractant. On the other hand, in the CBTP3 tumors, we have necrosis, and that might be actually the source of the neutrophils that come in that look a lot more like circulating neutrophils rather than tumor infiltrating neutrophils. And this really emphasizes the importance of looking at other rich phenotypes. And in fact, we're exploring both H&E stains and multiplex immunofluorescence with our genotypes in order to better understand these and other features. And so to summarize this final part, we built an engineered model of melanoma that were relevant mutations in different combinations on the background of primary human melanocytes. We showed that these have distinct phenotypes, hitting immortality with the first 3 mutations and then accruing in aggression and more pathways are hit. Single-cell RNA-seq allowed us to connect the mutations to cell intrinsic program changes in vitro and in vivo, and we show that these match those in the patient tumors, even down to the genetics that impacts them. And it also impacts the composition and gene programs in the microenvironment in animal models. And this can now be expanded, of course, to other mutation combinations, other readouts, other perturbations, and other tumor types. And so with this, I will conclude my talk. I talked to you about excluded tumors in melanoma and synovial sarcoma, and on two methodological approaches, one based on high-content single-cell RNA-seq-based screens in co-culture, and the second based on engineering cells with precise combinations of mutations. And I'll thank the people who led all of these projects. The melanoma exclusion program work was led by Livnat Jerby and Ben Izar when they were both postdocs in the lab. Livnat is now a professor at Stanford, and Ben is a professor in Columbia. And this started as a wonderful collaboration with Levi that I'm reunited with now in Genentech, and with Ellie Van Ellen, Dirk Schadendorf, and Bruce Johnson, all at the Dana-Farber. And the work on synovial sarcoma was also led by Livnat when she was a postdoc in the lab, together with a wonderful trainee in Mario Suva's lab, Cyril Neftel, and in collaboration with Mario Suva and Nico Ricci from Mario from the MGH, Nico Ricci from Lausanne. The work on perturbed site-seq was led by 4 amazing students and postdocs: Chris Frangi, a computational grad student; Johannes Melms, a postdoc in Ben Izar's lab. By this point, Ben had his own lab. and Pratikshya Sakor and Katie Geiger-Schuler are both experimental postdocs in my lab. And the work to build the melanoma was led by Eran Hodis, who started with this when he was a grad student in his MD PhD program. He's finished medical schools, he's doing his residency now, but the story is now finished, in collaboration with Elena Triglia, a computational postdoc, and John Kwan was the experimental research associate before he headed to med school, who worked the entire experimental system together with Eran. And this was really a heroic effort for both of them. And it was a wonderful collaboration with Levi, with Martin Mim, and with Labib Zaka. And with this, I will conclude and would be happy to take any questions.
Great talk, Aviv. And I want to say that there's several chat boxes left about how amazing your talk is, so I'm not going over these for you.
Thank you.
So I'll start with the first question by Enrico Pozzo regarding your first chapter on the melanoma. Impressive data. Is there any reason why abemaciclib has been used instead of other CD4/6 inhibitors? Thanks.
Oh, I wish I had a good answer to this, because it wasn't ideal, probably the ideal choice. It was the available choice. That was it. We wanted to do the work. We wanted to do it quickly. We had an hypothesis. We had access to abemaciclib, so we just went with it. In retrospect, we know that we might have done better had we chosen another CDK4/6 inhibitor, but the truth is also the result held. The other slight consideration was that we were inspired by beautiful work from Jin Zhao, who looked already at the effect of CDK4/6 inhibitor, and they used, I believe, abemaciclib not palbociclib and so on, on T cell biology in the tumors, and we were very anxious to understand whether what we're seeing is T cell mediated or not. So the killing is T cell mediated, but the effect depends on RB competence in the malignant cells, and that's how we knew that it was actually validation of this pathway. Excellent question. And I get it occasionally, because I know now that we could have done a little better.
Yeah, the second question is related to the synovial sarcoma story you presented. You show combined HDAC and CD4/6 inhibitors lead to increased T cell activation and killing. Do these T cells show improved tumor infiltration as well, or is the enhanced tumor killing occur at the leading edge of the tumor?
Oh, this was an in vitro co-culture system. So to my understanding, and I'm not a synovial sarcoma expert, but some of my collaborators are, animal models for synovial sarcoma are not great. And I would be more than happy to be disproved on that, but that was my understanding of the consensus in the field, that they have a lot of limitations. So we were limited to doing a co-culture system rather than an ideal situation where we would be able to do it at least in an animal model. And one of the reasons that we're developing, have put and will continue to put so much effort at these more patient-derived engineered models, like the ones that we've built for melanoma and the ones that we would hope to build for other tumor types, is that I think that, and as well as the use of things like organoids and so on, is that it opens additional paths for tumors where you don't really have animal models, and sometimes, which is not the case in synovial sarcoma, where you don't even have cell lines at all. So I've worked in our work, for example, on some of the brain tumors, like certain low-grade gliomas. There are, as I've learned, no great models. And as a result, the work on patient tumors like single-cell RNA-seq was really illuminating, because there was very little that one could tell besides from direct work from patient tumors. But then you hit a real roadblock on experimental studies to test hypotheses. So we're trying to push that envelope now.
Great. There's another follow-up questions about synovial sarcoma. So the HDAC and CDK4/6 inhibitors, their effect is partial. Have you tried to include DMT3A inhibitor to the combo to potentially increase the anti-tumor effect even further?
Yeah, it's a very good question. One point that I didn't make in the talk, but that is actually in the melanoma study that was published in 2018. So for the DNMT3 enthusiasts, it's there, is that we actually saw in those perturbation experiments with CDK4/6 inhibitors and in the co-culture system, we already saw that there was an effect on DNMT3. So our hypothesis, maybe correctly, maybe incorrectly, was that there might be a mediation through, rather than that we would be hitting a whole additional pathway and get a synergistic effect. But the truth is also that we didn't try. That we went a certain distance. We were very concerned about piling on too many medicines and starting just to hit into plain toxicity, because there is also a toxicity. I actually didn't show the slide, although I have one. It's in the paper. There are toxicity effects as well. So we're trying to stay in the place where we're impacting the program and are not just killing the cells ourselves with the overload of treatment.
Great, Jan. Another question from Lisa Span. Thanks for this interesting talk. I just have a more general question. It was already shown that there is a sex difference in immunotherapy for cancer like melanoma. Do you also detect these differences within your data, and how did you include them maybe in your analysis?
So we could not in our study. So the studies that we did, the early melanoma, in a sense, it kind of got the best end and the worst end of the deal. So when we started working on single-cell RNA-seq, our very first study, which I believe is actually the first one for solid tumors, was in glioblastoma. And so we developed kind of a line of work together with Mario Suva that I am personally incredibly privileged to have had the chance to participate in on gliomas. And when we wanted to move out of gliomas, in parallel, we started collaborating with Levi Garraway on melanoma. This had a huge benefit in letting us really hit the ground running. We could show what you could learn about tumors and so on. But it also means melanoma was the first. And because it was the first, this was in the early days, you know, where every dissociation was a research project in its own right, where we had to figure out how to move the samples through the hospital. All sorts of things like that played into it. We didn't have droplets yet. We were developing Dropseq in parallel to doing these projects, the ones that fed the very first part of the study. And so we had wonderful, wonderful and extremely committed collaborators. But the one thing we had to let go of was the idea that we would design a very careful cohort. Because if we had done that, we would have had to wait a pretty long period of time. And that's very detrimental to developing new approaches. So we parsed it out, right? In melanoma, we focused on moving fast. And in other areas, we focused on designing a lot. Where, for example, we have a CRC study, colorectal cancer study, that we have just posted on the bioRxiv maybe a couple of months ago in collaboration with Nir Cohen and many colleagues in the Dana-Farber and at the MGH and the Brigham, where we said, let's do a well-designed study where we could ask exactly those kinds of questions. But in melanoma, we can't even go back. The way that Livnat managed to eke out the exclusion program out of that study, I have to say kudos to her on showing what you can do with small data. So having said that, there is now a Human Tumor Atlas Network, which is an NCI-funded project, and I was actually one of the PIs on one of the centers. I'm now in Genentech, so I'm not, but I'm still watch from the side and cheerlead, and some of my lab members are still wrapping up projects where, for example, in melanoma, we are doing a study that would allow answering that question. So I think a lot more is going to come. And the other side of that question is, now where I am currently in Genentech, these are exactly the kinds of questions that we can answer in a much more elaborate way because of the clinical trials and the samples that have been archived as part of those clinical trials well before single-cell RNA-seq even existed necessarily.
Great. So, right, we are now at 10 p.m., which is the time for the closing of the official event. And I have to thank Dr. Greff for giving us a wonderful talk. And I also want to say that she has graciously agreed to stay on for an extended session. So we have a lot more questions that we can seek her expert review with us. So those of you who want to stay on, please stay on, as there will be more exciting discussions in the next half an hour that we can take advantage of time Aviv had with us. So, and those who want to sign off, and thank you all so much for attending this seminar. Okay, so let's start with the unofficial extended section now. What we have, the next question is, did you observe cross-link between genetic mutation and epigenetic alteration in tumors? This is from Jia Li.
Yeah, it's a great question. So in two parts, the work in the melanoma cells at the very end, that's one of our goals. We actually have a couple of collaborations that are going to allow us to look at that in a very mechanistic way, because we have these beautifully engineered mutations, and we can look at epigenetic changes at a very high resolution in a control system. where it's easier.
Mhm.
The work that we've done in colorectal cancer, even though it's not in this study because it was just too much for one study, but the way the cohort for colorectal cancer was set up and the samples were archived was in order to allow us to have a full multi-layered atlas. So there's the tumors are whole exome or whole genome sequenced. We have single cell RNA-seq. We will have single cell ATAC-seq. We have bulk chromatin, which actually came from this cohort and is already published in a study in collaboration with Brad Bernstein's group that was in Cell, like in the last very few months. There's microbiome data, all the, immune characterization data, and several different types of spatial data. So a portion, a slice of it is in the study that we've just recently posted that really focused on the RNA, and a tiny bit of genetics is there, and a tiny bit of spatial is there. And it was already a little bit of a tome. The paper was long and there was so much to say and so on, but we feel that that actually was a great starting point for us because it gave characterizations of the tumors. We understand how they're organized. We understand something about the niches that the cells are forming. And now we can connect it both to genetics and to epigenetics, as well as to other features of these tumors. And the study was made in this way from the start, because there we did have the benefit of starting with a plan rather than also paving the road while we were walking on it.
Great. Yeah, the next question is very interesting from KK Yan. In the last part of your talk, what do you expect in the inferred program if the mutations are engineered in different order?
That is something, okay, you want to know the original plan? I can't believe we were that naive. We were going to do a lot of combinations in different orders. So we learned some things in that project. The first was that until you hit the third mutation with TERT, these are extremely difficult experiments to do. The TERT was really heroic because these cells are not immortalized. You have them for a short amount of time. You have to do an edit and an edit on top of an edit and so on. The second thing that we learned, which is actually extremely hopeful, so it's the flip side of it, is that you don't have to do any selection. These mutations confer advantages. So the cells basically select themselves with a mutation. You don't add anything. There's no extra marker. There was no antibiotics involved, nothing. It's just a mutation itself driving it. And because of that, I believe that we can actually move to our second approach, which was our original first approach, which is to throw multiples of these at once at the cells. Now that we understand these kinds of, we have the reagents now, right? We understand which guides work. There were a lot of things we had to understand, but to throw them and let the mutations land in their own order, rather than kind of manually order them into this tree. But we definitely had to do it for this round because it was already excruciatingly difficult for very, very talented people to get the basics to work. And I think if we did not allow ourselves the ordering, we would be in trouble. If you ask me for my guess, I would say it depends. I think for some things it probably matters, but for many, many things, I have a feeling that it matters less than we would want to think. That a lot of what the mutations do is that they relax the cells in terms of the space of possibilities that they can explore. And with that relaxation, adding the next one, given the strength of these kinds of mutations, let cells get to the next space of possibilities, even if the route they go there is different. But that's a complete guess. I have no basis for my guess except to think so. I will allude also to the fact that in cell reprogramming, a very vibrant field for more than a decade, much more than a decade, probably 15 years, we have learned probably that order matters less than we thought. But just dumping multiple things at once, as long as they're very strong, can jump. We're not actually recapitulating history. But that does not mean that it won't matter in the clinical course. So you could end up with the same endpoint in a dish or even in a mouse, and it could look like the human tumor. But in the clinical course, having this combination at this moment in time versus that combination another moment in time can actually matter. I don't know.
And so, Aviv, I'll ask a personal question here. So Peter Van Loo and Paul Spellman has published a paper this last year on inferring the kind of somatic alteration events across all human tumors. I think melanoma must be one of the kind of the disease they have. It might be interesting that, to check the orders that really worked in the cell line model
Yeah.
actually confer with the inferences they obtained from
Yeah,
the
I
tumor
think that's
sample.
it's a great idea also because we have some failed paths. It's not that everything worked. And we also know which failed paths failed in animals, which we don't really go into. I definitely didn't talk about it here because we are never certain if it failed experimentally or it failed in a foundational way, right? Is the biological result or is an experimental stumbling block? So we're careful about negative results, like everyone is. But I would say that that when Eran made his choices, he definitely made them based on what we had for melanoma genome analysis, in which he's a great expert because he's one of the two people in Levi's lab who discovered the TERT mutation and did some other things. It's something that was very near and dear to his heart, trying to understand this question both of mutation combinations and mutational orders. But there was only so much guessing that we could do. So we feel we're kind of on the right track in terms of the main pathways. It's pretty reasonable, the first three, that they tend to be early. That's supported by literature and so on. And I think it's fascinating to consider comparing to the Spellman paper. Yeah.
Okay. The next question is from Max Franco. Do the cell programs depend on the KO temporarily? You know, it's the same question. That is, you have 5 guides that are used in one particular order, but I imagine there isn't one particular order of mutations in patients. So if you have to, it's the same line, the random line.
It's the same answer too. I don't really know. What I do know is that when we have the situation of the 5 and the 4 that are nested in them, we have seen case, and I actually tried to mention it when I was talking. It's in the case of the neutrophil infiltration that we can have what we would think of as right. We have 2 ways in which we have a neutrophil infiltration, and then we have a 5-way mutation setup, right, which is more aggressive and has each of the 4 nested in it, and we don't see neutrophil infiltration. So it's definitely a case where it's not the order of mutations. That's the only one where I can actually say that. But in general, on these things, I think our understanding of combinatorics in biology is pretty lame. I say that about me personally, not a complaint about anyone else. That's because there is a lot of combinations. One of the things we've worked on and work on very, very heavily in our group is to try to get to ways above this, beyond this combinatorial hurdle where we could actually make good combinatorial predictions. But we're just not there yet to say. For this system, for sure, I'm not going to say. Yeah.
Great. The next question is from Francisco Sanchez Rivera.
Francisco, hi.
Fantastic talk.
Okay, just to say, this is someone I've known since I taught a class at MIT when he was a grad student. So I
Great, yeah, I'm glad we got that. It's about synovial sarcoma. What do you think dictates the most bimodal patterns of SS18 SSX oncogenic program and cellular state in synovial sarcoma cells? Did you by any chance look at co-binding patterns of KDM2B and CPG methylation? Is it possible that this heterogeneity in the oncogenic program involves a de-differentiation lineage switch in these cells driven by the fusion?
Okay, there's a tons of questions there. The main person to address them to is Sigal Kadosh, just to be fair. Okay, so Sigal's lab focuses a lot on this specific question, and I think has interesting answers to it, some of which are published and some which might not be. I actually don't even know which ones are on the published side or not, so I'm going to be very cautious in answering. But I definitely think that there is a mechanistic explanation from the SS18 SSX fusion protein side that explain some of the programs that we see. Hang on 1 second. Apparently, I wasn't charging my laptop and it's about to go to sleep. That would not be a good idea. It's bad. And the battery runs out really quickly when you're presenting. So I just want to say to Francisco, I actually don't know the answer up front in the very generalized sense. But if you look at the data, even in the synovial sarcoma paper, Livnat dug into it a little bit because we did have this differentiation program, and there is a control that goes on differentiation, and there is a control that goes on this exclusion program, and there's a way in which the cell cycle is actually subservient. So the cell cycle is not directly controlled, with the exception of CDK6 and CDK6 and CCND1 or 2, but otherwise the control is mediated for the cell cycle, but there is definitely a differentiation control. On the differentiation side, which again, I didn't talk about it all in the talk, but was in the paper itself, we actually describe it more. On the differentiation side, there's two parts to how you think about the differentiation program. One are the two differentiated states. There's an epi-like and a mesenchymal-like, but there's kind of also being just not differentiated, which is somewhat related to also being more mesenchymal. So it's a little complex there. And we at first thought that we had a stronger statement to make, and then we decided that we're not certain of it, and we were not going to validate it enough to make the stronger statement on how the differentiation behaves. But the exclusion program is not differentiation. It's independent of it, and these are two separate processes. Whether there is an interaction term between them goes back to the answer I gave on the order of mutations. We don't know.
Okay, the next question is from Ken Chen. Hi, Ken. How important a stroma microenvironment is to immunologically cold tumor? Has in vivo work done yet in the synovial sarcoma portion of the talk?
We have not done any in vivo work with synovial sarcoma for the reason that I described, that at least my colleagues who are actual experts in this felt that the in vivo models were not just complex, but actually were not seen in the community as, I would say, as appropriately predictive, especially on clinical and therapeutic applications, which was our focus area. And that highlights a challenge, right? What do you do next?
Mm-hmm.
Which for me feels very palpable, that all of a sudden you can't just hide behind, I understood enough biology and wrote a paper, which is very compelling in its own right. But it's a different feeling, I think, for people in St. Jude's, that you feel the same, that you want to also see the next step, because you see the patient.
Exactly.
They're not some hypothetical thing you write about in your paper.
Yeah.
So I feel the same.
Great. Yeah. So the next question is from Andrew Navarez. After third promoter mutation introduction in the last vignette, the EMT program was turned on. Yet in the mouse model, there was not much metastasis. Since there's a clonal evolution in the metastasis to enrich for the third promoter mutation, any insight why?
No, so we don't know why, but it is a very correct. So two things. First of all, the EMT program turns on, and then it turns off. You know what? Let me try and share my screen again. This will be easier with the actual slides. Give me 1 second. I'll have to go back just a little bit. It's almost the end of the talk. I think it's here. Okay, you can see how the EMT program is really high. It's really strong in the center here. That's actually the transition. You see, these are the CBT cells. These are still CBT cells with telomerase, the red ones. But you can see that there's still some of it in this and this, which are actually the next cells in the editing tree. They're the next mutation, and then it basically dies out. So the really highly metastatic cells are not EMT. They're after the EMT. I mean, after. I just mean they're no longer expressing the EMT. They're with mutations that they have these additional mutations, and you don't see the EMT again. These cells are indeed not truly strongly metastatic, and you need a whole pile of additional things to get to aggressive metastasis in this model, just to be fair. So the reasons for that have to do then with other mutations. This program of EMT is simply not enough. I think it's actually interesting. Now, at the same time, this is melanoma. Melanoma and EMT is a different relationship than how people think about EMT in standard epithelial tumors, just to be fair. EMT is something that you hear a lot about in other tumor types. In melanoma, it's always looked a little off. And then if you look at the in vivo results, you see that the EMT is actually in a different phenotype, in a different genotype than the one that you have in vitro. It's not in the CBT cells are not actually going to do much. They can't really go far, CBT cells. So you end up with an EMT, but it's only in the CBTP cells, and only in some of them, not in all of them. You see, these are all the different P10s, all of these, but only some of them actually express the EMT. So it's a more complex relationship than the one that we saw in the in vitro results, highlighting even in NSG mice the difference between patients.
Okay, so Aviv, you may keep this slide on because the next question.
Next question. I'll just keep my slides on. The reason I went to the slides is that I only see I have to move this little window around so I don't hide my own slides. Yeah.
So the next question is from Orest Blashchuk. Please comment on the E and N cadherin expression in your melanoma model.
Oh my God, off the top of my head, I don't know. I just don't know. I'm more than happy to answer this question by email by actually checking.
Okay.
We might have something, I'm looking quickly because this has actual genes on it, but nothing that rings a bell on
about the cadherin.
Yeah, about cadherins. And I'm more than happy to check. It's a very easy check, but it would require an email to me because I will forget otherwise.
Okay, another question from Madame Mohan. In your study, when you stratify patients into responders, partial responders, and non-responders to immunotherapy, do you happen to investigate if there are germline correlates of code or hot programs in addition to what you observed? Okay, so that's a great question. And hi, Madan. It's nice to meet people, to hear names that I'm familiar with. The answer is that we did not. But it is the same, and the reason for that we didn't do it in our study is that this cohort was actually the basis of a completely separate study and just generously shared with us the cohort before they published their study. And that's a study that Ellie and Dirk led and that was published in Nature Medicine, I think this year, either this year or late 2019, who knows, the pandemic kind of mixes up all dates in one's brain. And they try to look at as many correlates as possible. I would still say that the 112 patients doing this association with a germline without an hypothesis is not going to survive. There's no way. Okay, but there are emerging studies in the community, I would say broadly, that are sufficiently powered now to start answering this. You're just not going to do it with 100 patients. For human genetics, I would say it would have to be pretty extreme to do it with 100 patients. I shouldn't say categorically it won't succeed, but pretty extreme. But there's interesting ways in which you can start teasing out signals, and I think we're going to start seeing studies like these, although not from my own lab. Not my own lab, at least we haven't done that. But Eli Helen is the right person to ask. He's really, I think, committed a substantial amount of effort and dedication to try and address these kinds of questions. Yeah, will be the type of the question that a lot of people in the therapy will be interested in knowing, so that you can pre-discover these germline association and kind of I am with you. The patient. Yeah, I'm with you. And for me, that's one of the reasons that working in the setting that I work now, you don't hit that block, right? You can do all of those things that I've shown in this talk are the kinds of things that we do in Genentech, but you don't have to stop at some point, and then you have this abrupt wall and you just can't pass through it to that next question, which is so obvious to us as a scientist. That's the next thing to ask. But in order to ask that, you need a clinical trial, you need a large patient cohort, you need the ability to make a small molecule or to use a small molecule, the ability to make an antibody. I mean, I happen to give an example that was really driven by small molecules, but it can be an antibody, a cell therapy. And so you want to have the ability to carry these things all the way through. I am really sorry that in this talk I can't, but that for me was a real motivation. I think it's very similar to the people who work in St. Jude's, that they have a similar feeling and a similar ability then to carry to the other side in the context of childhood cancers. So Aviv, the last question is from your friend, Francisco again. That's an extra one. You were my student. That's always, not my lab student, but my student in a class at MIT, and he gets extra. Yeah, so is it fair to state that p53 mutation act as an inflection point in the melanoma genetic series that explodes the number of the cellular states accessible for the evolving tumor? So there's two different kinds that work, okay? You can see that both P10 and P53,
they lead the CBTs in two different directions. And it's quite interesting because you have the CBTP and then P3, which is with P10 and P53, and then the A, which is with APC. So you can take P10 and add to it P53 or APC, and you would go even further and further, or you can take P53 and go down and go that route, or you can just add APC directly and you do an even bigger jump. So I would say P53 is one of several possibilities of how to get to that expanded phenotypic space. But it's not the only one possibility to get there. And I think that's actually consistent with what we know from patients. The other thing to point out, since you ask about P53, is that the P53 tumors do show the expected genetic instability, do show the expected genetic instability that you would expect once you knock out P53. And it's not in the same way that happens in the other tumors. It was just not in my slides. I only gave it as a short vignette at the end. So I didn't dig into everything, but we actually whole genome sequence those cells. So we know, and we see some interesting events that happen in P53 tumors, but don't happen otherwise in other tumors, which are what we would expect to happen in the genomes of P53 mutated cells. But it's not
Great answer. Yeah, so
not the only way. Cancer, don't fight evolution. That's hard. You need evolution to fight evolution. That's the immune system. That's what I learned from my adventures in single cells.
well,
No, it's not. It's a little extreme what I said, but I think it's a really, really tough battle with cancer. And when you see these things and you think about the space and power of mutations, and you combine that with the flexibility of cells, you understand why our therapies work when they do and why they don't. You don't understand the fine details, but you kind of look at the magnitude of it, which was well known to the clinicians and the drug developers and the oncologists. And it's humbling. I think it's humbling, but it's also very motivating to try and do better.
Great.
I think we answered a lot of questions. This was
yes,
so much fun
so
for me.
I have to say that, it was amazing that we almost spend all the 30 minutes of the extended section. And thank you, Aviv, so much again for the wonderful talk. And also for those who stayed on for this very engaging extended section. And we learned so much more from your talk during this very, engaged discussion with Aviv. And let's just give us a round of applause for Aviv, and we can use our, I cannot show these modules too. Thank you so much. Really appreciate you spending time with us in the lecture and in this extended talk. And people also wondering if the seminar will be online.
It will be available within a week of the lecture. Thanks for asking.
Yeah. That's my understanding as well.
Okay, I just want to share one last comment from Suraj and Pauli. We are waving hands and clapping.
So I'm waving. It was so nice to do this talk, and it felt so nice to be among friends and old acquaintances and new ones. So I appreciate you joining.
Bye, everyone.
Bye, everyone. Have a great day.
Thank you.